Trastuzumab emtansine, antibody-drug conjugate in treatment of human epidermal growth factor receptor 2 positive metastatic breast cancer. The human epidermal growth factor receptor 2, HER2-targeted therapies have improved clinical outcomes for patients at any stage of HER2-positive breast cancer, BC. Trastuzumab, a monoclonal antibody that targets the HER2 receptor on BC cells, showed improved survival in metastatic BC, MBC. However, resistance to therapy arises in the majority of patients with advanced disease. Antibody-drug conjugate, ADC, is a relatively new development to deliver cytotoxic drugs specifically to cancer cells. Trastuzumab emtansine, TDM1, is a HER2-targeted ADC, composed of trastuzumab, a stable ether linker, and the potent cytotoxic agent, emtansine, DM1, derivative of emtansine. TDM1 has been approved for use in patients with MBC who have failed prior therapy with trastuzumab and taxane. Dose-finding phase I study established the maximum tolerated dose at 3.6 mg/kg every 3 weeks. Phase 1 and 2 studies of TDM1 have shown clinical activity and a favorable safety profile in HER2-positive MBC patients. The Phase 3 randomized EMILIA and TR3RESA trials demonstrated that TDM1 significantly improves progression-free and overall survival in pretreated HER2-positive MBC patients. Nausea and fatigue are most commonly reported adverse drug reactions with TDM1 and cardiac toxicity comparable with standard of care therapies. The drug is well tolerated in most patients, with a predictable pharmacokinetic profile and minimal systemic exposure to free cytotoxic DM1. TDM1 has emerged as an effective therapeutic option for the management of patients with HER2-positive MBC.